So before we get started talking about gluconeogenesis, let's start by setting the stage. This predominantly happens in the liver, so we're talking about hepatocytes. Inside every hepatocyte, there's a mitochondria, which is called a mitochondrion if there's just one. Each hepatocyte is also going to have an endoplasmic reticulum that is used to transport stuff out of the cell, but we'll talk about that a little bit later. And next to every hepatocyte, there's going to be some kind of blood vessel that's able to take what the hepatocyte makes to the rest of the body. Now let's talk about the actual steps of gluconeogenesis. We start with pyruvate, which has to enter the mitochondrion. Once it's inside the mitochondrion, it's converted to oxaloacetate, abbreviated to OAA. This step requires ATP, which is converted to ADP in the process. Next, oxaloacetate is converted into malate, which creates NAD+ using NADH. These are similar to what's happening in the citric acid cycle, where you've got malate going to oxaloacetate, using NAD+ to create NADH. So, to prevent this malate from entering the citric acid cycle, it has to leave the mitochondria and enters the cytosol, where it's converted to oxaloacetate, and now that NADH gets regenerated. Back in the cytosol, the oxaloacetate is now going to get converted into phospholinopyruvate, which is abbreviated to PEP. The enzyme that's used is called PEPCK, and it requires the use of GTP. Some other reactions occur that take PEP to 2-phosphoglycerate and then 3-phosphoglycerate, but those aren't that important. The next reaction we really care about is 3-phosphoglycerate becoming 1,3-bisphosphoglycerate, which is abbreviated to 1,3-BPG. This requires the use of ATP to get that extra phosphate group on the BPG. Next, the extra phosphate group is going to leave, and G3P, or glyceraldehyde 3-phosphate, is going to be made. The enzyme that's used to make this is called glyceraldehyde 3-phosphate dehydrogenase. And NAD+ is going to be made by using up NADH. Inorganic phosphate's also made, but that's not drawn here. G3P is used to make DHAP, and those combine together to make fructose 1,6-bisphosphate, then converted to fructose 6-phosphate, and this is the rate-limiting step of gluconeogenesis. Because fructose 1,6-bisphosphate can be abbreviated F16BP, the enzyme that does this is called F16BPase. The last step of gluconeogenesis converts fructose 6-phosphate to glucose 6-phosphate. Glucose 6-phosphate can then enter the endoplasmic reticulum, which we talked about before, where it is converted into glucose. This glucose is packaged into a vesicle inside the endoplasmic reticulum, where it ends up leaving the hepatocyte, entering the blood, and able to go to the rest of the body.